Good evening, everybody. Can those of you that are in the back of the room, can you hear me okay? Okay, I'm getting a thumbs up. If at any point you can't, just give me those thumbs up and I'll assume I need to talk louder, hopefully, or I'll just appreciate the encouragement. I'm so glad that y'all are here tonight. Thank you for trusting me and trusting HudsonAlpha with an evening of your time. And this talk we do every year around this time, and it is one of my favorite nights of the year, where I get to stand up here and tell you guys some stories that we've collected that we think are just incredibly cool, and I'm excited to get to bring you into them, and hopefully you think they are just as fascinating and cool as I do. I imagine many of you, like me, maybe you get emails with roundups of information. Maybe it's the news and current events. Maybe it's some other hobby, right? But you get these collections of really interesting things that are happening. And so tonight, that's what I'm going to do for you guys. So you can kind of sit back, and I'm going to take you on a journey through science that was discovered last year. So cutting edge, way too new to be in anybody's textbook that they're learning in school. This is science that if you rewind a year ago, we didn't know about as a scientific field that hadn't been published. And all of these stories that I'm going to tell you tonight come straight out of this resource right here. That you might have picked up a little magnet when you came in that looks like this, just shrunk down, Honey, I Shrunk the Kids style. But this is our annual biotechnology guidebook. And we publish a new one of these every year around this time as a resource for you guys as the community, but also as a resource for teachers and students in classrooms to be able to bring cutting edge science in, right? To be able to know and talk about the stories that are happening right now in research labs here at HudsonAlpha and all across the globe. But being able to read about them in a way that you can understand. So we, throughout the year, scour and read lots and lots of really technical papers. We pick out our favorite stories, and then we write about them in simple ways for folks to read and understand and get excited about what's happening in science today and what's going to be happening in science tomorrow. So this is last year's version. The one for 2025 is literally at the printers right now, but it's actually online. You scan that QR code, that'll take you to this year's version, which has the stories we're going to talk about tonight. So that's the backstory, but I am super excited to get to just story tell with you 6 of my favorite stories from this year's guidebook. So let's get started. The first story I want to tell you is about how DNA can tell us a lot about ourselves. Sometimes we use genetic testing to think about what might happen in the future. But this first story I'm going to tell you is about how DNA can tell us stories about the past. DNA is a very resilient molecule. It can stick around for a really long time. Thousands and thousands of years if it is stored well, right? If it's not exposed to a lot of heat and humidity and sunlight, DNA can last for a really long time. There's a story that I'm not going to tell you about tonight, but you can read about in the guidebook about discovering and studying the DNA from woolly mammoth skin that they found that's been buried under ice for tons and tons of time. But the story I do want to tell you has to do with, if my clicker will work, takes us back to 79 AD and the eruption of Mount Vesuvius. This is a painting, clearly, not an actual photograph from 79 AD. But this is a painting that was that's entitled "The Last Day of Pompeii," that was painted in the 1800s. And I imagine many of you in this room know something about this eruption of Mount Vesuvius. I see some nods. That happened in 79 AD. This was a massive volcanic eruption where these clouds and plumes of molten rock and ash and gases went 20 miles up in the air at crazy speeds, and it went on kind of in cycles for 2 days. Massive eruption, massive destruction occurred, and so towns like Pompeii ended up completely covered in ash, including all the people that unfortunately were in Pompeii that day. All of those individuals ended up covered in ash and lost. But back in the 1800s, as archaeologists were uncovering the areas of Pompeii and trying to learn about what was happening way back when this volcanic eruption happened, they discovered these cavities inside the, at this point, solidified ash, right? And so they found cavities and, come back. And these cavities had a human form because the organic matter of the bodies decomposed, but it left a void in that solidified ash. And so very astutely, these archaeologists decided to fill, as they found these voids, they filled them with plaster. And so what you have are these human forms of plaster that perfectly mimics that open, that space that was left by that body. More than 100 of these exist that have been found and filled with plaster. When they first started doing this, they moved them to special museums, and then at some point they decided to actually leave them where they were found. And so these are filled with plaster, but inside some of them, you know what's there? Good answer. DNA, yes. There's bits of bone that are left and are in the plaster. And so you might be wondering, this is supposed to be science from last year, not a long time ago. But what happened last year, or was published last year, is as the archaeologists and the people who are caretaking these casts, as they are doing work on them and fixing them up and preserving them, as they find bone, they're allowing for DNA to be extracted from that bone. And so what was published and made public last year was some information about 5 individuals who were victims of the Pompeii, the Mount Vesuvius explosion. And it has kind of reshaped some of the things we thought we knew about these individuals that were victims. So you can tell a lot. You can kind of see how much these look like humans, but if you were to look really close, you would actually see facial expressions. You would be able to see what kind of jewelry they had on them, what they were wearing, what they were carrying at the point they met their demise. And so from that, people start making stories. They start making assumptions about how people are related to each other. There's a story about 2 of the people that they were able to do DNA testing on, that it was an adult carrying a child. And for a long time, the assumption has been this is a mother carrying her child, partly because the adult had on a really intricate bracelet. Turns out they're both male. And they're not related to each other at all. Now, I really wish I could satisfy the end of that story and tell you how they did know each other. We have no idea. This is more of a process of exclusion than inclusion. So that was one of the stories. That adult and child were found alongside 2 others. And so the assumption was this is mom and child and maybe 2 more people, and this was a family. Nope, all males, and none of them are related to each other, at least back to 3 generations. There's another story of 2 individuals that were trapped in an embrace. And this, if you had asked people until last year, they would have said the going theory is that these are sisters, or this is a mother and a daughter. Now, we only have DNA out of one of the 2 individuals at this point. What do you think the punchline is? It's a male. Yeah, all 5 of the individuals that they have DNA from and they tested were all males. And so we know at least one of the 2 individuals that were embracing each other were males. And then finally, the other piece of kind of fascinating new knowledge that came from this study was looking at the DNA from an ancestral background. And all of the individuals that they were able to test and get information from were all from Eastern Mediterranean areas and were immigrants into Pompeii. And so it can tell you something about the makeup of the people who were living in Pompeii at 79 AD. I hope that this is just scratching the surface. And it's really exciting to think about what DNA is hiding out there, whether it's in woolly mammoth in the Arctic or it's in Pompeii, that can help us better understand the past. So from Pompeii to parasites. If you hear the word parasites, you really get excited, right? This is you get positive feelings. Well, I hope maybe I'll change that by the end of the next 5 to 7 minutes. So I'm going to tell you a story about parasites that is actually positive. I might gross you out in the meantime. So the story here starts with a problem, and that problem for science is this issue of the blood-brain barrier. So our brains are really important. We can all agree there. And the flow of blood and oxygen through our brain is really important. And our body wants to preserve the function of our brain. And so there is this structure, this blood-brain barrier that is wrapped around all of those capillaries and blood flow in our brain. That's job, it's a gatekeeper. It wants to let the right stuff in, it wants to let the bad stuff out, and it wants to keep the bad stuff away. And so if we were to take a cross-section, this is a kind of complicated picture, but this is like cutting one of the capillaries, one of your blood vessels in your brain in half. And we're looking at it from the side, where you have the capillary, the blood flowing in the middle, and then you've got a whole set of cells around it that are working together to keep your brain safe. It creates physical barriers where it just flat out things can't get through. It's very tight. There's also chemicals and enzymes that are working that can open gates at the right time and let nutrients in, or open gates at the right time and let toxins out of the brain. But these are very controlled mechanisms for good reason. However, this can be a problem for medicines, and you have people who we need to get something into their brain to help with a particular disease, and that blood-brain barrier isn't always kind to that. It often blocks. More than 90% of medications are not going to cross that blood-brain barrier. So this is a massive problem when you're trying to treat diseases that cause symptoms in the brain. You know what can get in your brain? I mean, parasites, yeah. In particular, this story, I would like to introduce you to this particular parasite called Toxoplasmosis gondii. From here on out, I'm going to call it Toxo. The rest is a mouthful. Toxo is, like I said, it's a parasite that has absolutely no trouble getting into our brains. In fact, if we go back to this last slide, the way it crosses the blood-brain barrier is it just comes right up here and infects these endothelial cells and jumps right in them, okay? No problem. Toxo is a fascinating parasite that I would like to spend just a minute and explain to you the fascinating life cycle of Toxoplasmosis gondii. The only definitive host for this particular parasite are members of the domestic cat family and their relatives. Okay, that is the natural host for Toxo. And so the life cycle up here that doesn't include humans goes something like this. The cat poops out spores or oocytes into nature that is then eaten, ingested by birds, rodents, pigs, sheep, you name it, things that eat nature. They ingest it, and when those oocytes get inside of those intermediate hosts, they go out and they can invade and they take up residence inside the muscles and the organs of those animals. The brain, skeletal muscle, right? And so what do cats do to rodents and birds? They eat them, yeah. See, it just goes around in a little circle, right? Cat, rodent, cat, rodent. And so that is the kind of typical lifestyle. Humans come into the mix in a couple of different ways. Humans can get infected with Toxo by eating uncooked meat from an animal that had ingested those spores and they turn into cysts inside the muscle. Humans can also get infected by doing things like cleaning a cat box. Yeah. We think about a third of the world's population of humans is infected with Toxo. You're welcome. You're welcome for that. But for humans, most of the time, that doesn't matter. For humans, it does not cause symptoms. It's just an asymptomatic thing that we live with, knowing that these things might be in our bodies. There are some situations, if someone is immune compromised, that they can have symptoms, like even in their brain from this, but it's rare. The biggest issue with Toxo in humans is when mothers get infected for the first time, or like new infection while they're pregnant. And so if you've ever heard the advice that pregnant women should not clean out cat boxes, it's not just they want a break. That it can actually cause birth defects in that developing child for those new infections. So that's the fascinating life cycle of Toxo. But the reason that we're talking about this at all is because toxoplasmosis has these really interesting ways that it can deliver proteins into the cells that it is infecting, including brain cells. So here's a schematic. This is a picture of a toxoplasmosis parasite that's invading a particular cell. And so it comes up to it, and there's two different processes here. As it's coming up on a cell, it kind of sits on the top and injects a side of it into the cell, and it has these structures called rhopteries that are able to inject proteins into that cell. These are proteins that the parasite has instructions for, right? It's got the DNA for making a protein, and then the authors of this paper call it kiss and spit, where it doesn't even have to be inside the cell. It kind of just nestles on top and spits protein into the cell. But then when it continues to invade the cell and get inside, there's another process through a different structure inside the parasite called a dense granule that is able to, through a different mechanism, transport proteins from these granules can leave and head out into the cell as well. So there's two different ways that toxo can put proteins that it's made out into the cell. And that's how it's putting its own proteins out there that help it survive and replicate. But what if we had a way to give the toxo instructions for delivering a protein that we want to get into those cells? So that's what the scientists have been studying, and that's what was published this last year. That a group of scientists, they bioengineered Toxoplasmosis gondii to produce therapeutic proteins. So these are proteins that would not naturally be made by the parasite, but they're proteins that could be beneficial to humans in certain situations. So they were specifically picking proteins that are important in human brain disorders. They went ahead and picked those to do in this study. And so the way that they did this was they went in and they identified areas in the toxo genome, in the toxo DNA, that coded for proteins that the scientists knew were secreted or exported through those dense granules. So the dense granules, this gene right here that codes for the GRA16 protein, we know that that protein is excreted through dense granules. So what did they do? They inserted instructions for a therapeutic protein next to it. They created a fusion gene. So that would make a protein that does both of these things. Big protein, two functions. And they did the same thing for a protein that we know is secreted through that kiss and spit method through those rhopteries. Okay? So that gene is toxophilin. And so they, again, attached instructions for a therapeutic protein right there next to it. So we know that if these things are going to get secreted, they'll get secreted through those pathways. And then they tested it. And here's what they found. It worked. In the lab, right? I will preface this that this was, they were not going and knowingly infecting people with toxo. But it worked that these parasites were able to successfully make and secrete to pass off these proteins, these fusion proteins. And they actually could do both of them simultaneously. So they tested this in a couple of ways. First, you can see the progression of the study here. They started with human skin cells in the laboratory, and they proved that it worked. That after 24 hours, more than half of the cells, those human skin cells, were infected by the toxo, and they had the protein inside of them. But those are skin cells. We're wanting to get this into the brain. And so they did a similar study with human brain cells, again in a laboratory. And for this study, I mentioned right here that they were studying this therapeutic protein called MECP2. MECP2 is the gene and the protein associated with it that's associated with Rett syndrome, which is a very significant neurodevelopmental disorder in humans. So they were studying that gene in particular and showed that after 24 hours, the neurons in the lab had achieved more than 50% of what normal function of that protein. And then finally, they tested organoids. So these are cells that kind of work together as a little kind of organ in the lab. And here they were testing, well, it's one thing to deliver this fusion protein, but does it work? Does that protein actually do what you would expect it to do, or is it broken in some way? And they showed that in both situations, that the proteins functioned like you would expect them to. So it delivered a functioning, working copy of those proteins. Exciting, huh? Yeah. Now, this is not like you're not going to run out and get a prescription for a parasite from your physician. Clearly, there's some more work to be done before this becomes a valid, viable way to treat neurologic disorders. But it's a fascinating possibility of if there were to be a way to develop non-virulent, right, strains of toxo. At no point are we going to be intentionally giving people toxo in its natural virulent form. But if you could find a way to make it not virulent, but make it deliver stuff we want it to, leveraging its innate ability to get into brain cells is pretty cool. So parasites can be positive. Maybe. But don't change the litter box if you're pregnant. All right, my next story for you. If you think back to biology class, now some people in the room, you might not have to think back too far to be in biology class. Looking down here. Some of us, it was a few more years ago. But I suspect many of us in biology class maybe saw pictures that looked like this, right? These are cells under a microscope that are undergoing division, right? Mitosis, where cells are dividing, and there's all these different phases. Maybe you had to learn and identify these different phases in a textbook. But at the crux, what we have always believed is true is when cells are dividing, you start with one cell, and it goes through all of these steps, and you end up with two daughter cells that are just like the parent. This is normal cell division, like what's happening in our skin cells. This is not creation of egg and sperm. That's a whole other story. But this is the way that typical cell division works. You start with one and you end up with two. Most of the time, we thought, but now we have discovered a unique situation that is happening in all of our mouths. It's not a parasite this time. It's a bacteria. So much better. But there is a bacteria that harbors, that it lives in our mouths, in our gum lines, that has the innate ability to, let's just watch it go for a minute. This is a bacteria. It's growing. It's a very long, stranded bacteria. Yeah. It's great. It keeps going. This is a bacteria called Corynebacterium matrucciati. I think that's how you say that. And this is a bacteria that we have now discovered that is able to simultaneously divide not into two daughter cells, but as many as 14 at the same time. Yeah. So what's happening inside this bacteria as it is growing out and out and out, it's fascinating. It is actually able to copy its DNA over and over and over again without splitting. And so when it is that really long, right before it splits, it's got as many as 14 individual copies of its DNA. And then somehow, we're still trying to figure out the how, it sends signals to break up into all of those individual daughter cells, each with its own set of DNA. You're so welcome. Fascinating, right? And I love these stories where it takes what we thought we knew, what we thought we knew as a solid fact of science, and it says, but wait, there's exceptions to that rule, because that's what science is, where we're constantly learning new things, and we're kind of bucking those long-held hypotheses and expanding our knowledge of what's happening in the world around us. And so this provides some insight into how some bacteria can divide in really interesting ways, can divide really rapidly and create things like biofilms. That's what's happening on your teeth and gums, or on slippery rocks out in the creek, where bacteria are growing together in a film and communicating with one another and trying to suck up as much nutrients and space as they can. But it makes you wonder what else is out there, right? That does this, or maybe there's some even other more unique ways that bacteria or other types of organisms are dividing. Trying. Okay. So the next story I want to tell you, so we're going to go from bacteria over to plants, and some work that was done that helps us better understand how plants grow and how certain plants grow really, really tall, really, really fast. So this right here, this is a picture of a poplar tree. And if you might know this about poplar trees, is they grow really tall, really quickly. If you see a young poplar tree, it might just go straight up, right? They invest in growing really tall, really fast to get up above the canopy before they ever really bother putting out branches. And so we know that about poplar trees, and there's some poplar trees that grow even faster than others. And this past year, this is not HudsonAlpha's work, but scientists, so the broader we, discovered a specific recipe in the poplar, specifically the cottonwood poplar, that is contributing to that really fast cell growth. Guess what they called that new gene that they discovered? Booster. That makes sense, right? Yeah. So this is a gene that was discovered. The gene is not new. The gene's been around forever, but they discovered it in the poplar that boosts plant growth. And the way that booster works, this is about to be a really busy slide, but for people that are students or teachers that are watching, this might mean something to you, because I think we teach about photosynthesis in the classroom, right? So the booster gene, what it does is it increases the amount of rubisco in the plant. Rubisco is a protein. It's actually what we believe to be the most abundant protein on planet Earth, because rubisco is an enzyme that starts photosynthesis. So how plants make energy and grow. When you think about how many plants are on planet Earth and how much rubisco is in all of those plants leading to photosynthesis, there's a lot of rubisco around. And so this gene and the protein it encodes for, it increases rubisco, which in turn increases photosynthesis, which leads to faster plant growth. This is a picture. These are poplar trees that are less than a year old. And these are two scientists that were working on studying the effects of this particular protein. And you can appreciate the difference in size of these two poplar plants. The one on the left is about 20% taller. They were planted the same day. But the one on the right has less booster expression. That protein is not working as well as the protein is working on the left. The other fascinating thing about the story is, and part of why this gene was just discovered and what it does, is that this gene is a fusion, it's a fusion gene inside the poplar tree. We were just talking about fusion genes with the parasites, where scientists in the lab created a fusion gene. This is a natural fusion gene that's inside these poplars that has bits of DNA from a couple of different genes. One is the natural rubisco, the natural booster gene in the poplar, but there's some bits of DNA that are added to it that lead to its ability to make more rubisco. There's portions of this gene that have come from bacteria that we know reside around the roots of poplar trees, as well as some parts of the DNA of this gene that came from the ant genome that lives in the bark of the poplar trees. So at some point over history, bits of those other organisms' genomes have made their way into the poplar genome in this particular spot, and has, by chance, led to that gene having increased production of rubisco. Yeah. Very cool. So this, we know this about poplar trees. This helps us explain what's going on with poplar trees. But then that naturally leads to the question of what's next. Is there a way to use this information then through biotechnology and applying it to solve some other problems? Are there applications to other types of crops that it would be really great if we could make them grow faster to produce more food? Perhaps there's an opportunity to boost them, right, pun intended, with booster. So that is boosting plant growth with booster. The next story I'm going to tell you about is related to Alzheimer's disease. So heading back to humans, and a problem that is near and dear to many of our hearts and minds. I don't think I need to give a huge introduction to what Alzheimer's disease is, because of how important it is and how many people are affected by it. Something like 1 in 10 adults over 65 is affected with Alzheimer's disease. And so it goes without saying that there is a lot of research being done in figuring out the causes and better diagnostics and treatments for Alzheimer's, including a lot of work here at HudsonAlpha that is focused around the genetics of Alzheimer's. But the story that I want to tell you is about a treatment that got FDA approval this last year for Alzheimer's disease. And part of why this is one of my favorite stories from the guidebook is for a long time we've told people that Alzheimer's disease is a non-actionable, non-treatable disorder, that you can treat symptoms, but there's not a lot available in terms of treating the underlying causes of what is leading to Alzheimer's disease. And this is an example, and it's one of several that are starting to do exactly that. So in Alzheimer's disease, this is a picture of what your brain is supposed to look like and what your neurons are supposed to look like. But in Alzheimer's disease, there's a number of things that are going awry. In particular, there are 2 different types of proteins. One is called amyloid plaques. That's shown up here as this A beta plaques. And the other are tau. And these are 2 different proteins that we all naturally make in our bodies, but usually we're able to break them apart and clear them. But in Alzheimer's disease, these proteins don't get cleared by the body. They hang around when they shouldn't, and they get all tangled up. We often call them tangles either for the amyloid, they get tangled up in between neurons, and the tau, they're tangled up kind of inside those neurons itself. And those tangles mess with the neurons and make them not work and ultimately die. And that then leads to the shrinkage of areas of the brain and leads to the symptoms of Alzheimer's disease, from those early symptoms of memory loss all the way up to the more advanced symptoms that affect language and movement and all of that. The medication that I want to draw your attention to that was FDA approved this past year, so this is way further down the chain than the parasites. This is actually approved and ready for human use. This is a medication that targets those amyloid plaques, those tangles of proteins in between the neurons. This is a medication called Kisunla, and what this medication does is it's an infusion. I believe it goes into the arm, but it's an infusion that patients take about once a month. And what it does, and this medicine is able to cross that blood-brain barrier, not through a parasite. It actually is able to get brought in through a channel. It's able to, a receptor is able to mediate moving it into the brain. But this medication is essentially antibodies that goes out and knows how to find and attach themselves to those amyloid plaques in the brain as well as throughout the body. So the antibodies attached to them can bring them together and then ultimately alerts and brings in the person's own immune system to get rid of them, to clear them. And so the clinical trials that were done showed that this worked pretty effectively. In fact, people were eventually able, they took them off the medication because their plaque levels were dropped into the normal range. There was nothing left there to clear, and so they would come off of the medication. So what happened in these clinical trials that led to the approval? Now, this is not a wonder drug that can halt the progression of Alzheimer's or reverse the progression of Alzheimer's. But what they saw in patients with Alzheimer's who took this medication was a slowing of the progression. So the decline, so this chart right here is showing the functional impacts of Alzheimer's disease, and the black line is the placebo, people that were not taking this medicine, and the orange line were people who were. And so there was a slowing of progression and a prolonged quality of life. They found in the study that the people who benefited the most were people that were earlier on in their Alzheimer's diagnosis. And so this is a type of medication that's going to have the best effect if you're able to get a diagnosis early and start this kind of medication. But you can see the decline still happens, at least in part because this medication is targeting the amyloid proteins, but it's not doing anything to tau, that other protein set of protein tangles. That you would imagine that an ideal approach is going to have to attack this from more than one direction to have an even bigger impact on Alzheimer's progression. But this is definitely a big step forward towards treatments in Alzheimer's. There's a number of other treatments that are kind of hitting the market and doing different things too. It's a really exciting time to be watching that space. This particular medication, though, is not a magic bullet. It also comes with not an insignificant price tag. You can imagine that's the way things go, right? New medications are expensive and often initially not covered by insurance. And so for patients, there's some weighing that has to happen in terms of risk and benefit. But it's definitely an exciting step in the right direction for a disease that affects a lot of people. All right, here's my final story for the night. And I would like this time to introduce you to a tiny little organism called a tardigrade, or a water bear. Isn't he cute? It looks like you just want to like squish him. Maybe it's the original Squishmallow. You have to have a 7-year-old at home anyway. Although this is way, way, way zoomed in. So these water bears, or tardigrades, are about 0.5 millimeter in size. So they're little, they're tiny, tiny, but you actually can see them with a standard microscope, right? This is not that tiny. But water bears were discovered back in the 1700s by a German zoologist who gave them their name water bear because they were cute and they looked like a bear. And then a little bit later, an Italian scientist came along and gave them a more technical name called tardigrade, which means slow steppers or slow walkers. Because they do kind of move in a sloth-like way. But tardigrades, or water bears, these are aquatic creatures. They like to live in areas like moss, and they are able to use that little snout. It's called a bucopharyngeal area that is able to extract, like suck out nutrients from microorganisms and from plants. I could watch him all day. But the other really cool thing about water bears, in addition to their cuteness, is the fact that they are well known for being able to survive just about anything. Now, we don't consider them extremophiles because they don't like to be in inhospitable environments. They don't thrive there, but they can survive. So they can survive hundreds of degrees below zero, hundreds of degrees above zero. They can withstand more than 1,000 times a lethal dose of radiation for a human. They've been taken to space and exposed to all kinds of stuff. They've been shot out of guns and survived. Yeah. They're really, really resilient little creatures. And we know some of why. So they also really, like I said, they're aquatic creatures, so they like to stay in wet environments. And if they dry out too much, they actually have this other form called a ton that they turn into, where they kind of dry out. And but as soon as they get back in a happy, moist environment, they go back to their cute little self over here and move on with life. But this doesn't explain all of their resiliency, their ability to switch forms, and it certainly doesn't explain the resiliency to crazy amounts of radiation. And so the story and the study in the guidebook this year was a study that was done specifically trying to answer the question of how do they withstand that 1,000 times human lethal dose of radiation. For this study, they took a multi-omics approach, and by that I mean they didn't just study the DNA of the water bears, they also studied the RNA and the protein. And all of these are important because you've got your recipe in the DNA. That is the water bear's recipe. But how it uses its DNA is it turns pieces of that DNA into RNA, which codes for proteins. And so by looking at the RNA and the proteins at a particular snapshot in time, it'll tell you how is it using its DNA. What genes are turned on, which ones are turned off. And you can compare water bears that have not been exposed to crazy amounts of radiation to the ones that have, and see what's different, right? How are they using their DNA differently when they are exposed to radiation? And that's exactly what they did. So they sequenced the DNA of these water bears, and fun fact, they actually discovered a brand new species of water bears by way of doing the study. But they looked at the DNA, and the DNA is largely stable over time. But then they looked at the proteome, that's the proteins, and they looked at the transcriptome, that's the RNA, for water bears that got no radiation, water bears that got a little bit of radiation, and water bears that got a lot of radiation. And through that, the scientists discovered more than 2,800 genes that were different. That when exposed to radiation, that those genes are being used differently. They're either being turned, it's like a volume control, right? They're either being turned up or they're being turned down. We're not going to sit here and go through the implications of 2,800 genes right now. I'll be here all night. I'm going to tell you about two of them real quick, though. One of the genes that they found that is different in the water bears after radiation is this gene called DODA1. And this is a gene that codes for an enzyme that's really important in helping organisms produce betalains. Betalains are a pigment that you often find in plants and fungi, and apparently water bears. But what betalains do is they are really important for controlling inflammation and cell damage, right? You can imagine that turning this up after radiation or during radiation would be a good thing. And that's exactly what happened over here. So these charts are showing the amount of this gene that's being made, so the amount of RNA related to this gene and the amount of protein. With no radiation, it's super tiny. With a little radiation, it's turned up a little bit, maybe. But then with a lot of radiation, the amount of this protein goes way, way up. So perhaps this is part of that puzzle. Also of interest, this gene, this DODA1 gene, is really, really similar to a gene found in bacteria. So kind of like that booster story a few back where DNA from ants and bacteria found its way into the poplar genome. This is likely inside the water bear genome because of this same process called horizontal gene transfer, where DNA is moving between organisms, and in this case from bacteria to the water bears, but had a very positive effect. The other gene that I wanted to highlight for you is a gene that is not from another organism. This is a gene that, to our knowledge, is unique to water bears. And this is a gene called TRID1. And what this gene does is it encodes a protein that helps repair, so fixing, of breaks in the DNA. Especially what is set up here, the double-stranded breaks, where both sides of a DNA molecule are broken, or bad breaks. This particular protein that this gene codes for help repair that. Do you know what causes double-stranded breaks? A number of things cause it, but radiation causes a significant amount of breaks inside our DNA. Perhaps, not perhaps, emphatically, this is playing some roles in helping fix those breaks, which could very much be a big part of the resilience of the species, and something that is unique to them and why they are uniquely able to withstand radiation. Interesting, right? And they're just so cute. If you're having a bad day, just Google water bears. So those are 6 of, I don't know, 40 or 50 stories in this year's guidebook. Like I said, there's a story about woolly mammoth DNA. There's a story about a tiny, tiny fern that has now broken the record of having the largest genome of any organism. There's several stories in this year's guidebook about how scientists are using AI and machine learning to help make sense of massive amounts of data. There's all kinds of good stuff in the guidebook. And like I said, this one is hot off the presses. It's actually not off the physical presses, but is online for your perusal. As we wrap up tonight, I wanted to draw attention to this logo that's been floating around that is our newly updated HudsonAlpha Alumni Association program. Some of you might have heard about HudsonAlpha's Alumni Association. You don't have to have a cap and gown to be in it. But it's our way of coming alongside people that have been a part of our programs and continuing to stay connected. And, oh, it went away. And the program is getting kind of a rebrand and a refresh in 2025, where now everyone who comes to one of our programs is officially in the Alumni Association, just automatically. Welcome. Yeah, it's so easy. But it's also a way for us to, for individuals to come alongside HudsonAlpha's education mission and support what we do for the community, but also with teachers and students and all of the programs and resources that we provide. And so I'm bringing this up because tomorrow an email is going to hit your inboxes with that new logo and some information about it and about the different levels that you're automatically in it, but that there are some levels that you can sign up for that give some extra benefits and provides extra support to us. So I wanted to just draw attention to that. For folks that maybe signed up for the Alumni Association a few months ago, don't worry, you're grandfathered into this. You'll get an email tomorrow. And for those who might be watching this after tonight, if there is a QR code on this slide right here, if you want to go learn more and be a part of what we do in HudsonAlpha education. But it is such a joy to get to see you guys face to face at these events. And the Alumni Association is just a way for us to stay in touch in between when we're able to be together in person. But thank you guys for being here tonight. And before we completely wrap, if you've been to these before, you know I take selfies. So smile. Okay. With that, we will close. I hope that we'll see you back at many of HudsonAlpha's other events throughout the year. The next one of these events with me up on stage will be this fall, where we're going to dig into this really fascinating topic called polygenic risk scores. It's a mouthful. We'll deal with it in September together.